Hi, everyone. So nice to see you all virtually here today. I'm Justin Colacino. I'm faculty in the School of Public Health. I'm one of the PIs of the Single Cell Spatial Analysis program. And today I'm going to tell you just a little bit about generating single cell omics data with the 10x Genomics Chromium device, which we have available at the Advanced Genomics Core here at the University of Michigan. And so it should come as no surprise to the folks in this audience that being able to generate and understand omics profiles of single cells has led to a revolution in biology, right? Where we can start with healthy tissues or diseased tissues, break these down into single cells, look at these omics profiles, and then really generate these in-depth cell type maps of both healthy and pathological tissue, looking at tissue homeostasis, looking at development, looking at disease processes, giving us the ability to search within cell types for things like stochasticity of gene response, the gene networks that are regulating cell types, or even looking at cell-specific allele expression patterns. We can also look between cell types, right? Identifying biomarkers which predict cell type identity. We can look for transcriptional or post-transcriptional differences, which are driving differences in cell types. And then we can start to look between tissues as well, right? Looking at cell type compositions between healthy and disease or stressed and unstressed conditions. And then even looking at altered transcription and matched cell types. Really, there's so much data that we can generate through these single cell experiments that it's just providing so many new insights into how biology is functioning. And the technique I'm going to tell you a little bit more about today at the U of M Advanced Genomics Core is this 10x Genomics Chromium device. And this is a droplet-based sequencing method that really takes advantage of these 10x barcoded gel beads. And so these gel beads, each of these has a little DNA oligo attached to the gel bead, and the oligo on each of the beads is different. So that helps differentiate the beads. And how this device works is these beads are flowed through in a microfluidic device, and then your single cell suspension is flowed through there too. These guys match up in little droplets, and then we can collect those gel bead cell droplets shown here in the picture. And then we're gonna do an in situ reverse transcription reaction where we're gonna lyse the cells, and then we're gonna create cDNA from the RNA, and then that cDNA is gonna incorporate those oligos. And then so those bead-specific oligos, and then all of the same RNAs from each individual cell get tagged with the same oligos. We remove the pool oil. We're left with this pool of barcoded cDNA, which we can then take to our Illumina sequencer and look at. And so one of the advantages of this Chromium technique is that we can profile both viable single cells or single nuclei. And so many of you have probably tried this technique using viable cells, right? Where you have to start with a single cell suspension, and this could either be, a trypsinized cell culture, this could be blood cells, or this could be cells from a tissue which you've had to enzymatically and physically digest, right? To generate a healthy, viable single cell suspension. If anytime you're dissociating a tissue, it's going to require a tissue-specific dissociation protocol, and these are going to require freshly collected tissues. There's also options to use nuclei isolation techniques and profile individual nuclei. So here we could take fresh or frozen tissue, dissociate these, isolate the nuclei, and then profile the RNA or the epigenetic patterns within those nuclei, meaning that we can leverage already existing specimens. Many of you are probably like me, we have a freezer just full of experimental specimens that have been frozen that may be waiting to be leveraged in these single cell techniques. And so with the Chromium here at U of M, we have a few different options at our disposal. We have gene expression. This is sort of like the classic method where we're doing 3' whole transcriptome profiling. We have ATAC-seq, where this is single cell epigenomic profiling, looking at regions of open chromatin. 10x has also released this cool multi-omic platform where you can do simultaneous ATAC-seq and gene expression analysis. So here we're doing simultaneous profiling of both mRNA expression and chromatin accessibility in the same cell, really giving you an idea about how epigenetic regulation might be driving single cell level gene expression. And then those of you who are interested in the immune system would probably be excited about this immune profiling platform that's been released. You can get whole transcriptome gene expression with additional modules, including cell surface protein expression using oligo-tagged antibodies. So if you have a set of antibodies that you've typically been using to differentiate cell types, say in a flow cytometry experiment, you can take those antibodies and attach an oligo. And when you incubate your cells with those oligo-tagged antibodies, the cells that are tagged with those antibodies will then have that oligo show up in your sequencing data. So that's like very cool. For folks interested in T cell receptor and B cell receptor recombination, there's full length TCR and BCR sequencing. And then there's also the opportunity to screen for antigen specificity with that same feature barcoding concept where we take barcoded antigens instead, and then can look for where those barcodes show up to tell which cells might be binding those antigens. And so you might wonder what a typical single cell data analysis pipeline looks like after all of this data comes back from the sequencer, right? So we get a series of FASTQ reads. Remember, these are going to be barcoded by each cell. We're going to undergo some QA and QC on that read quality, and then sort each cell out based on those cell barcodes, right? We can then trim the reads from those cell barcodes. We can map those reads to the genome, and then we can count the number of reads that align per gene, per cell, and you get one of these feature matrices where you have cells and genes. And this is what the 10x Genomics Cell Ranger platform will give you. You'll have this matrix at your disposal. After this, we like to do some QA and QC, do some filtering of low quality cells and doublets, look for batch effects, and then you can really jump in and analyze, right? You can generate these really cool unbiased clustering maps, these T-SNE or UMAP plots that we love. You can look at differential expression between cell types or across treatments. And then we can do neat experiments like pseudo-temporal ordering. If we think that we have a differentiation trajectory in our cells, we can really order these cells within the context of that trajectory. So a lot of like really neat analyses we can do. And so I just want to highlight a couple of ways that you can enhance your experimental designs that have recently become available. And one of these is called CellPlex. And the CellPlex technique from 10x Genomics allows you now to start to pool samples together. This lets us look at more biological replicates or more treatments in a single reaction. And so how this works is that we take our individual samples, and then we're going to label the cells in each sample with an oligo-tagged lipid. And then once we've got our oligo-tagged cells, samples, we can then pool these guys together and then go through that same process where we're going to encapsulate the cells, do the DNA amplification. And then when it comes time to analyze our data, we first look for that sample level barcode and then that cell level barcode, and we can start to mix up to 12 samples together. With this CellPlex technique in a single library prep. So this can save a lot of money and also save a lot of experimental headaches down the road. Another way that you can sort of start to combine samples together, do pooling, and reduce the potential for batch effects is through what we call genetic multiplexing. So if you're using samples that come from just genetically distinct individuals, you can also pool these together for processing prior to running it through the 10x Chromium device. And what we do is we look in that sequencing data and we can call genetic polymorphisms. And then based on those polymorphisms on each cell, you can link what cell that came back to from the individual. There's some really nice advantages here because it lets you identify doublets because those cells would have combined genotypes. And then there's multiple bioinformatics tools that are available for this purpose, including dMuxlet, which was developed here at U of M. And so when we're thinking about how do we design a study for 10x Genomics, there's a lot of things you want to take into account. This is something I'd be happy to chat with over email, but think about what are the source of cells that you're using. Do you want to look at purified cell populations or whole tissue? Do you want to do single cell or single nuclei? Are you interested in RNA-seq, attack, both? Immune profiling, how many biological replicates do you want to run? How much variation do you expect in your system? Are you going to profile deep, where you get a lot of reads in fewer cells, or shallow, where you get fewer reads in a lot of cells? And then can you pool? Can you use the CellPlex method? Or could you use this genetic multiplexing to save some money on library prep and also have some experimental advantages in reducing batch effects? And so I'll stop there. Thank you all for your attention. If you do have any questions about this, feel free to email me, colacino@umich.edu. Otherwise, thank you very much for your time.